Thank you for the introduction. I'm with the group of Robert Reben based in Bern. And this was done in collaboration with the group of Claudia Vasco and especially with Emily Coppa. So islet transplantation is one possible treatment for type 1 diabetes. However, a high amount of islets is needed for one transplantation, which is why xenogenic islet transplantation would be such a promising solution. However, a big hurdle to overcome is the occurrence of an instant blood-mediated inflammatory reaction, so-called IPMIR. And what happens is that islet cells express tissue factor on their surface. And tissue factor leads to activation of the coagulation cascade with formation of a thrombus and occlusion of vessels. There is also attraction of immune cells leading to the inflammation. Another player that is important in IPMIR next to the coagulation cascade and the immune cells is the complement system. And we were now wondering which complement components could play a role in IPMIR. For this, I had the opportunity to work with material from humanized mice. So these mice have human myeloid lineage cells, and they have been transplanted intrahepatically with porcine islet cells. We wanted, therefore, to study the reaction of the human immune system to porcine islet cells in a mouse model. So these mice have human immune cells, but we didn't know if they have human complement. Therefore, our specific aim was to analyze the human complement deposition on porcine islet cells in livers of humanized mice. And what I found was deposition of human C1q in the liver of humanized mice that had received porcine islet cell transplantation. You can see it in the picture on the left in red. I furthermore looked for other complement components, so the human C3b, C5b29, C6, as well as Murine C3, but I found no deposition. I also didn't find any deposition of human antibodies. So from this, I can say that there is deposition of human C1q in the liver of humanized mice. One possible explanation for this is that C1q is actually synthesized by myeloid lineage cells, and these cells in the mouse model are human, whereas other complement components are synthesized mainly by the liver, and the liver in this mouse model is not humanized. So I can say that C1q might possibly play a role in IPMIR, but we are still investigating this. And if you would like to know more about my project, please come visit me at my poster number 250. Thank you for your attention.